morning, dear students. In the last class, we have learned about the DNA replication method in prokaryotes. And in that video, I have told you that in the next video, we will learn something about the properties of DNA polymerase enzyme. So let's start with that in today's section. That is structure and function of DNA polymerase. And first point that we need to learn is about the replisome. What is replisome? Replisome is nothing. It is the hollow enzyme form of DNA polymerase 3. It is a hollow enzyme form. That itself means that this DNA polymerase 3 is a multi-subunit enzyme. Its image we can see here. On looking to the image itself, we can understand that it consists of so many different subunits, and each subunits have its own catalytic role. So, this replisome can answer say 2 mark question for your exam. So please note that. Let's see some other important features of this DNA polymerase 3 enzyme. Among the different subunits that present in the DNA polymerase 3, they are present a non-catalytic subunit. Its name is beta clamp. Its name is beta clamp. And what is its significance? It helps the DNA polymerase to stay associated with the DNA template to complete the DNA replication. In what fashion this beta clamp is holding the DNA template that I will show you in this fashion. This DNA template will pass through that beta clamp in this way. So in the form of a ring, this beta clamp can help the DNA polymerase to stay associated with the DNA template to complete its replication. And that attachment is loose enough to allow the enzyme to move freely along the DNA template. And we know that replication happens in two strands, in the form of leading strand and the lagging strand. Then what about the mode of movement in leading strand and lagging strand with respect to the polymerase? In the leading strand, this polymerase can stay associated with the same beta clamp until finishing the replication of that strand. But in the case of lagging strand, it cannot do like that, because there, on completing one Okazaki fragment, it need to dissociate from that beta clamp and need to associate with another beta clamp that assembled in the next RNA primer. So that is the way how the polymerase moves on the lagging strand. Okay, then another non-catalytic subunit that present in the DNA polymerase 3 is the uh, clamp loader. What's its name? Its name is clamp loader, and it has a specific name also, that is wrench. What is its significance is that it is responsible to load the beta clamp to the DNA in the form of a ring and allow it to fit around the DNA. So that is wrench. Then let's understand some more points regarding other different types of DNA polymerase. I have already told you in the first section that 3 different types of DNA polymerase present. In prokaryotes, they are DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. So next, let's see about DNA polymerase 1. This DNA polymerase 1 is responsible to repair damaged part of DNA. And it is the same enzyme. It removes the RNA primer after the completion of DNA replication from each Okazaki fragment. And basically, this DNA polymerase have two main function. They are uh, polymerase function and exonuclease function. Exonuclease function, it can act in two directions, from 5 dash to 3 dash, as well as from 3 dash to 5 dash. So, polymerase activity is significant when means after removing that RNA primer part from each Okazaki fragment, they need to synthesize the DNA part. It is done by that DNA polymerase 1. I will show you later in the next slide. And that 5 dash to 3 dash and 3 dash to 5 dash exonuclease activity also, they are left with the DNA pol 1. Exonuclease activity means it can degrade, remove the deoxyribonucleotide, it can attack the deoxyribonucleotide and remove, that is exonuclease activity. And regarding that 5 dash to 3 dash, 5 dash to 3 dash exonuclease activity, it can act on both DNA and RNA. Clear? That 5 dash to 3 dash exonuclease activity of DNA pol, it can work on both DNA and RNA. That is another important special feature of this DNA polymerase 1. So next I am going to show how it removes the RNA primer and how it resynthesizes the daughter strand in place of the place where that RNA primer is removed. Let's see that. So this is two strands, and here we can see that RNA primer, this orange color also RNA primer. Magenta color is that replicated daughter strand portion. And the DNA polymerase 1 came, and it removes the RNA primers. RNA primers all are removed. Next thing, it need to synthesize. It synthesize the DNA portion in the space from where the RNA primers are removed. So that is what next happening here to that Okazaki fragment. That lost part means in that space, in that gap, the DNA strand, daughter DNA strand portion is synthesized by DNA polymerase 3. And let's see what about the ends. This will be the 5 dash end, and this will be the 3 dash end, and from that 3 dash end, this DNA polymerase 1 is adding the deoxyribonucleotides, and on reaching the 5 dash end of the next Okazaki fragment, the DNA polymerase 1 cannot join this 3 dash end with the 5 dash end of the next Okazaki fragment. There left a small gap. That gap is filled by another enzyme called DNA ligase. Okay, and I think these much points are clear to you. That's importance of DNA polymerase 1 and DNA ligase. Okay. Next is about DNA polymerase 2. What is its significance? It has some functions like 3 dash to 5 dash exonuclease activity. And we have seen that in the case of DNA polymerase 1, it has both 3 dash to 5 dash as well as 5 dash to 3 dash exonuclease activity. So, as in general, we can tell that all the three different types of DNA polymerase have 3 dash to 5 dash exonuclease activity. Exception that DNA polymerase 1 has 5 dash to 3 dash nuclease activity also. So, let's once more come back to the case of DNA polymerase 2. It has 3 dash to 5 dash exonuclease activity. Also, it can proofread. It can proofread and process what mismatches that are created by the DNA polymerase 3 while adding the nucleotides, deoxyribonucleotides. So that mismatches can be corrected by DNA polymerase 2, and also this enzyme can repair and correct interstrand cross links. Unwanted interstrand cross links also can be removed by this enzyme. Also, this DNA polymerase 2 has a primase activity. Primase activity, we know that because of primase activity, this DNA polymerase 2 can lay down an RNA strand against the DNA. Okay, so these are the functions of DNA polymerase 2. Exonuclease activity, proofreading, repairing, and primase activity. Okay. Then we need to understand how that fidelity or that quality of that DNA replication is attained. So let's see. First of all, the geometry based consideration. Geometry based consideration means as adenine pairs with thymine, that gives a perfect geometry for the DNA strand. And as guanine pairs with the cytosine, it gives the perfect geometry with regard to that distance between the atoms as well as the bond angles of the two bases. So considering the those aspects, what combination, which all combination give best geometry? Adenine thymine base pair as well as guanine cytosine base pair. So looking to or considering that identical or sorry that best geometry, the DNA polymerase 3 is binding or selecting an incoming nucleotide. It check whether it is having an incoming nucleotide is having a perfect or good geometry to pair with the base in the template strand. Is it clear? Suppose this is the template strand and suppose it is the base and this is the DNA polymerase 3 as this is the incoming nucleotide. Then this enzyme, it check the geometry of this incoming nucleotide, and as it find that it has a matching geometry with the thymine, then it allow this base pair with this thymine. So that is one method. It assures the fidelity or quality of DNA replication. Then second, you somehow think that an incorrect base is incorporated to the newly synthesizing or replicating DNA strand, then what will happen? That incorrect or mismatch incorporation left a free 3' single strand end. Free 3' single strand end. And that end give a conformational change. Give a conformational change to the enzyme, and because of that change, this incorrectly incorporated base become oriented to its exonuclease site of this DNA polymerase 3, and that exonuclease nuclease activity of this DNA polymerase 3 at the moment itself remove this incorrect base pair and select the correct one and form a pairing and create a pairing bonding with the base that present in the template strand. That is the second method. 3' to 5' exonuclease activity. Okay, then there exists a third method to assure the fidelity or accuracy of replication, that is mismatch repair. Somehow, even though that geometry level checking is there and also that exonuclease level correction is there, if those two levels has failed and somehow a wrong base got incorporated to the duplicating daughter strand, then their function this mismatch repair mechanism. this mismatch through this mismatch repair mechanism, that wrongly incorporated base is removed from the replicating DNA, daughter DNA strand. So, in a general sense, we can tell that which are the three main methods that assure the fidelity. First method is the accurate selection of nucleotide, and second method is the immediate proofreading, and the third is the post replication mismatch repair. So through all these three methods, the DNA replications, that fidelity or accuracy of replication is assured, or it is attained. So these are the three methods. Next, we are going to understand about how the termination happens in the case of prokaryote. We have learned that the replication begins at a point called origin, and from that point, there will be two replication forks, and that replication proceeds in this direction from this fork, and from this fork, the replication proceed in this direction. Okay, and this direction is clockwise, and this direction is anti-clockwise. Okay, and as these two replication, this two-way replication proceeds, it will meet somewhere here, and on meeting there, that marks the end of replication and resulted two daughter DNAs. And that is the simple idea, or simple explanation about how the termination happens in the case of prokaryotic DNA replication. But some more details we need to understand how the termination happen on reaching this point. That is made by a certain sequence that present in this point. Its name is TER sequence. So let's see about those sequence here. In this picture, we can see three TER sequences. Here also, in this side also, we can see several TER sequences, and at those sides there present some TER proteins also, and those TER proteins, I'm going to draw a single TER protein, and a single TER protein have two face, one a flat face and a pointed face. As the replication continues, we know that it is led by helicase, by unwinding the twisted DNA helix. So, as the helix moves through the DNA double strand, on reaching this phase of the TER protein, the helicase can easily pass through this TER protein. So, consider the helicase that is moving in this direction. As it proceeds, it is facing this phase of this TER protein. It will not face any hindrance, and it can continually move through all these TER proteins, and will reach here. And we need to understand that this point, if a helicase that is moving in this direction, On reaching this pointed phase, it will stop that helicase. It stalls that helicase. So that is going to happen here. As this helicase proceed, it can move through these proteins through here, and on reaching here it face the pointed face of the ter proteins on other side, and that helicase cannot move further, and the replication that proceed in this direction stops here. And in the similar way, the other helicase that is moving in this direction, unwinding the DNA, it can easily pass through all these TER proteins. And on reaching here, it again face the pointed face of this TER proteins, and this TER protein stop that helicase here, and that replication process also got stopped on reaching this point. So that is how the termination occurs in the case of DNA replication of prokaryotes. I think it is clear to you. With this, I'm going to finish today's section. Thank you.
I'm an icon, man. Ain't my girl about a 10. I should fuck her in a Benz.